Lysosome, another cell organelle, is very important, as you know that the recent research is saying that if your lysosome is very active and if you utilize your lysosome to kill your own cell, you will be cancer free, because you know that the affected cells, cancerous cells, all kind of cell can be digested itself automatically with the help of only one organelle, and its name is lysosome. So let's start how we can activate the lysosome and becomes a cancer free. So lysosome is an lysosomic organelle है, सेल organelle, बट माइंड इट, अब तक हमने जो भी सेल ऑर्गेनेली के बारे में बात किए हैं वो सारे डबल मेम्ब्रेन सेल ऑर्गेनेली थे. दैट इज योर एंडोप्लाज्मिक रेटिकुलम, your Golgi bodies, your plasma membrane, all were double membrane. But plasma membrane is not organelle, but it's a structure. But in this case, this plasma membrane, this organelle is a single membrane, single unit membrane structure. And you know that unit membrane structure single is present, another is your peroxisome. And non-membranous structure is ribosome and centrosome. So let's start the lysosome. Lysosome is a cell organelle which is responsible for digesting of your own cell. So ये क्या हो सकता है कि एक सेल ऑर्गेनेली जो सारे सेल को डैमेज कर सकता है, एक एटम बॉम्ब की तरह सेल में ही है, तो क्यों रखें उसे? Why it is stored in cell? It is actually a defense mechanism. Now recent research is saying that your lysosome is actually the protection shield, because sometimes if cell is working in a improper way, that it can develop cancer or it can be affected or modified or becomes mutated. So your lysosome can help you, but it has some criteria. What is actually the main criteria? Lysosome is actually a sac-like structure and is developed from Golgi apparatus. From where? Golgi body. Golgi body से तैयार होने के बाद ये कहाँ पे जाता है? ये जाता है Golgi body से तैयार होने से बाद ये प्राइमरी लाइसोजोम से सेकेंडरी लाइसोजोम बनता है. सेकेंडरी लाइसोजोम कैसे बनता है? ये प्राइमरी लाइसोजोम जुड़ के सेकेंडरी लाइसोजोम बनता है और उसके अंदर ठूस के भरा होता है low pH acid hydrolysis, all kind of hydrolysis. They are proteases, your lipase, carbohydrate digesting enzymes, all kind of enzymes. There are more than 50 types of enzymes are there inside the lysosome. So what is the function of this? मैंने बोल चुका हूँ कि the main function of is the destroying your own house to protect your body. So in case of plants, the lysosomes are not present like animal cells. वो जो vacuole होता है, वो vacuole में वो pH जो होता है, pH, ये जो इसका जो lysosomes का जो pH होता है, pH बहुत घटा हुआ होता है 5. ये 5 pH वाला, that is highly acidic. And this acid, if present inside your cell, all your organelle, all types of organelles will be non-functional, because its function needs a 7 pH. Now here lysosomes are, what are the different types? In case of fungi, lysosome is very high, but our body contain all cells, all is a lysosome, but only exception one is there. This is RBC, red blood cells. So now primary storage organelle, that is primary or storage organelle, that is called lysosome. Primary lysosome, secondary lysosome, that is heterophagosome, here the residual body, and autophagosome. So primary lysosome is a storage granule, is also called just forming, that is primary lysosome is just formed and the proteins and enzymes are just in the form of granules. They are stored and just stored and not in a functional form. After this, from primary, it is developed a secondary or heterophagosome. Heterophagosome is the functional, that is, it has a function to digest, okay, the organelles or any ergastric materials, mainly not organelle, the ergastric materials that is entered by accidentally inside the cell. So this part is primary storage granules and heterophagosome. Heterophagosome is mentioned. ये जो हेटेरोफेगोसम होता है, ये क्या होता है? हेटेरो मीन्स नॉट ऑटो, that is outside body material if entered inside your cell. It's called heterophagosome. They will be phagocyzed, that is, that will be killed by the lysosome. This is called heterophagosome. And a residual body, that I have mentioned, that residual body is a lysosome that has a debris, that is, after digestion some debris is present inside the lysosome. That lysosome is releases material outside the cell by exocytosis. You know that exocytosis, endocytosis. So, and autophagosome means they receive the material, intracellular material, that is cell organelle. Cell organelle जो होता है, that is Golgi body, endoplasmic reticulum, ribosome. These all material को खाने के लिए ये है autophagosome तैयार करता है. एक ही लाइसोजोम का डिफरेंट जो कैरेक्टर है वो है ये. क्या होता है? पहले ये जो लाइसोजोम है, ये प्राइमरी लाइसोजोम है, इसमें एनगल्फ करने की, डाइजेशन करने की उतना क्षमता नहीं होता है. और ये जो सेकेंडरी लाइसोजोम हो जाता है, तब ये क्या होता है? हेटेरोफेगोसम बन जाता है. बाहर का मटेरियल सारे गिल जाएंगे, सारे गिल के खा जाएंगे, निकल जाएंगे. ओके, देन रेसिडुअल बॉडी होता है. रेसिडुअल बॉडी एक्सोसाइटोसिस के लिए होता है. पहले बोल दो 3 टर्म: पिनोसाइटोसिस, फेगोसाइटोसिस, लिसिस एंड एक्सोसाइटोसिस. सेल ईटिंग, ड्रिंकिंग एंड vomiting. That is, pinocytosing is a cell drinking, phagocytosis is cell eating, that is solid material they will take, and exocytosis is cell vomiting. It will release the outside, release material outside. Then autophagosome, cell organelle. Autophagosome is its own cell organelle can be digested. So it is called suicidal bile because the total cell function will be disrupted. Now the intercellular digestion is 2 types: autophagy and heterophagosome. Intracellular, this heterophagosome, that is heterophagocytosis, that is means the outside material if entered inside the cell, the lysosome will be destroyed. Lysosome will destroy them. This is called heterophagy. And autophagy means if own cell organelle is to be digested. These are extracellular digestion is if the your body's unwanted material is developed, can be digested. Even if in embryo condition, if unwanted organ development will be started, it can be digested by the lysosome. So it is very important. Except RBC, in all body cell it is present. Now the cellular digestion means your suicidal bag. That is, I have mentioned that if cell is not proper functioning or defected or being affected by the any organisms, virus or bacteria, or it's automatically being converted into कैंसर सेल. अगर कैंसर सेल में कन्वर्ट हो गया और अफेक्टेड हो गया और दूसरा ऑर्गेनिज्म से, देन इट कैन बी किल्ड ऑटोमेटिकली बाय द लाइसोजोम बाय इट्स हाइड्रोलाइटिक एंजाइम्स. और एक बात ये लाइसोजोम के तैयार इम्पॉर्टेंट है, वो है इसका जो पीएच इतना लो होता है. मैंने मेंशन किया पीएच 5 होता है. क्यों होता है? क्योंकि इसमें एक्टिव ट्रांसपोर्ट सिस्टम है, दैट इज H+ आयन को बाहर से अंदर ठूस के भर के उसका पीएच घटा देता है, और इसके लिए ATP is needed. That ATP energy is there. So it's the active transport system is there. H+ ions inserted inside the lysosome. So that is the lysosomal pH being reduced. So you have to remember and take note each and every point, each and every term, so that you can answer these questions. Thank you.